The overall goal of this procedure is to identify new drugs that modulate GIRK channels. This is accomplished by first plating the AT-T20 cells in the 96-well plate. The second step is to dilute the test compounds using the Vazquez liquid plate handling system. Next, the diluted test compounds are applied to the cells using the Vazquez. The final step is to insert the 96-well plate into the Synergy 2 fluorescent plate reader and inject the control and somatostatin solutions into the wells. Ultimately, the fluorescent plate reader is used to show the compounds that alter the somatostatin-induced fluorescent signal and thus modulate the GIRK channels.
The advantage of this procedure over existing methods, such as patch clamp recording, is that a large number of compounds can be quickly screened to determine if they modulate GIRK channels.
The implications of this technique extend towards treatment of pain because compounds that activate the GIRK channel, which lead to inhibition of nociceptive transmission, can be identified.
Demonstrating the procedure for coating the 96-well plates and plating the cells is Maribel Vazquez, a student in my laboratory.
Begin this procedure by growing the pituitary AT-T20 cells in DMEM, supplemented with 10% horse serum at 37 degrees Celsius in a humidified atmosphere with 5% CO2. Maintain the culture by subculturing the cells every 5 to 7 days using a standard trypsinization procedure. Next, coat the wells of the black clear bottom 96-well plate with 50 microliters of poly-L-lysine. Allow the wells to dry for 30 minutes in the incubator. Then remove excess poly-L-lysine. Plate the cells in the 90